at the door. Don't run away, it's all me. Don't be afraid of what you can't see. Don't run away, it's only me. Don't be afraid of what you can't see. It's only me. With only me.
It's good to have you back.
Standing there alone, the ship is waiting, all systems are go. Are you sure? Control is not convinced, but the computer has the evidence. No need to abort. The countdown starts. Watching in a trance, the crew is certain, nothing left to chance. All is working, trying to relax. Up in the capsule, sent me up a drink. Jokes, Major Tom. The count goes on. 4, 3, 2, 1. Earth below us, drifting, falling, floating, weightless, calling. Second stage is cut. We're now in orbit. Stabilizers up. Running perfect. Starting to collect. Requested data. What will it affect when all is done? Thinks Major Tom. Back at ground control. There is a problem. Go to rockets full. Not responding. Hello, Major Tom. Are you receiving? Turn the thrusters on. We're standing by. There's no reply. 4, 3, 2, 1. Earth below us, drifting, falling, floating, weightless, calling. Across the stratosphere, a final message. Give my wife my love, then nothing more. Far beneath the ship, the world is mourning. They don't realize he's alive. No one understands, but Major Tom sees how the light commands. This is my home. I'm coming home. Earth below us, drifting, falling, floating, weightless, coming.
Hi, everybody. Welcome to Nerd Night. Yay! It's back. Folks, we've been doing Nerd Night for almost 10 years, and we took a long COVID break. And we got a couple going over the summer, and we had a couple in a few places, but this is our first time being back at our home base, our headquarters, our best friends live. Thank you, live. It is really, really nice to be back where we belong. So, my name is Sarah. I work at the Ann Arbor District Library, and I am your host for this evening. I'm very excited about the speakers that we have lined up for you tonight. Thank you for coming out and choosing to be in a dark bar and not in the first nice evening of the spring. Sorry about the conflict. We didn't know it was going to be nice. It's so rarely nice. I didn't know to plan. So I'm going to give you a little spiel about what Nerd Night is, and then I'm going to introduce our first speaker, and then we're going to get underway. So, Nerd Night. Tonight is our 79th Nerd Night. We have been doing it for a minute, but we weren't the first to have this brilliant idea. That would be Chris, who was in college in Boston all the way back in 2003. And Chris was studying evolutionary biology, and those studies would take him to Cameroon, where he would study creatures and write papers, stuff PhDs do, you know. Well, while he was there, he would study the indigo bird. This is a beautiful little bird. Look how cute that bird is. Got a little birdie mohawk. He's just a cute little bird. And he was fascinated by the story of this bird because as cute as the indigo bird looks, he was not cute in his heart of hearts. No, in fact, he was a jerk bird. And what he does, the birds, they lay their eggs in another bird's nest. And then those birds, when they're born, they are jerk birds by their very nature, and they murder the babies who belong in that nest. And then the parents, who are nice but dumb, raise the jerk bird instead, who go on to lay more eggs in other nests and kill more innocent baby birds, and the cycle continues. And it's horrible. But if you're an evolutionary biologist, it's pretty badass. And you're like, I got my paper right here. I'm going to be a PhD genius. And he was. But when he would come home, just like every, person who studies abroad ever, he would go to his favorite bar and he would tell his friends all about the amazing things he learned. And he would do it so often at his favorite bar that his favorite bartender was like, you have to stop. I've heard so much about this jerk bird. I can't handle it ever again. And Chris was like, come on, just one more time. And he was like, fine, invite everyone at once. Don't tell this story one by one by one. Get them all in. You can tell it one last time. And he did. And everyone was like, this is the best. We should do it all the time. So they do. And it's called Nerd Night. And now there's like 1 million of them. Look at all of that. I noticed there is not one in Cameroon. And that feels like a ripoff because they are integral to the story and the birth of Nerd Night. So anyway, yeah, you can go to Nerd Nights all over the place. My favorite one is right here in Ann Arbor, and that's because all of you are here. Aw, come on. That was so sweet. All right. I'd like to thank the Ann Arbor District Library, who sponsors us. I would like to thank Live again for having us and setting everything up. And I would like to thank all of you guys again for coming out. Seriously, it's awesome. We super appreciate it. It's good to have you. And now to introduce our speakers for this evening, we've got Emma Thornton-Kolbe. She will be telling you all about Fruit Fly Genome names. They are weirder than you thought, but she's always known, and she's ready to finally tell you. Leanne Sue will be telling us about propulsion stuff, and I can seriously tell you no more than that. I have looked at her slides 3 times. I can't wait to have them translated for me. And Kayla Zochowski is going to tell us all about our DNA and the past and the genetic code and the ways people are doing it wrong and the things you can find out and the things you can't find out, and it's total bullshit. But Kayla won't lie to you. She's going to tell you everything. So first up, please join me in giving a very warm Nerd Night welcome to Emma Thornton-Kolbe.
Okay, hello. I'm just gonna test that this works, and it does. That's very exciting. Thank you all for coming to Nerd Night. I'm really excited to tell you about Fruit Fly gene names, which maybe doesn't sound that exciting. I promise it is. So I'm gonna start off by getting a general temperature in the room. How do we feel about fruit flies? Like thumbs up, thumbs down, thumbs to the side. Okay, so there's some thumbs up, which is surprising. But I'm seeing a lot of thumbs down, some heckling. I'm ready for it. You're all wrong, or those of you who said thumbs down are. And I'm going to tell you why. So I've been working with fruit flies in the lab for the past 6 years, 2 years as a tech, and then the last 4 years here at the University of Michigan as a neuroscience graduate student. And I think they're great, mostly because they are not rodents. Rodents bite and they smell bad. And everyone who works with rodents tells me they've developed mouse allergies. It seems bad. Fruit flies are also great in that they're not rodents, and so I can take their brains out without wanting to pass out, which is a huge thing for me, a neuroscientist. I want to look at the brains. So this is a brain I've dissected, and those are some neurons that you can still kind of see in there. And over these past 6 years working with fruit flies, I've learned that the wonderful fruit fly community, all the scientists who work with fruit flies, have some fun when they name genes. And so tonight I'm going to tell you about some of those gene names and why they're called that, in the hopes that maybe you'll feel a little more positive about fruit flies before you squish them. I recognize they're an annoying pest. I too find them annoying, just in my house. I like them in the lab. All right. So just so we all start off on sort of the same page, I'm just going to let everyone know what a gene is. So this is a strand of DNA that I've drawn. It's a double helix. I've highlighted this little red bit. That little bit of DNA is a gene. I've written out the sequence. The sequence codes for a protein, which you can kind of think of as a tool that a cell would use. And so every cell in the body has a bunch of DNA stored in chromosomes, shown here. This is like technically a fly cell because flies only have 4 chromosomes. Humans have 23 pairs. Flies have 4 pairs. So all of the DNA is stored in there. So there's hundreds of thousands of genes stored in all these different chromosomes. But every cell isn't going to use every single gene. It's only going to make the tools it needs to be a cell. So cells of lots of different types make up human bodies. They make up fly bodies. That's supposed to be a fly. Kind of hard to draw. Right, so a brain cell is going to need different tools than a skin cell. A brain cell is going to want to make things, is going to want to use genes to make things like neurotransmitter receptors so that it can talk to other neurons and send messages across the body. A skin cell might want to make proteins to hold it to other skin cells so it creates a solid, barrier between the outside world and the inside world. And so you can imagine that if any of these changes in the gene are going to be reflected in the tools the cell has available, and the cell is going to do a little bit different of a job, and that's going to change the overall phenotype of the organism, right? So the way the organism looks or the way it behaves. And so, right, genes are clearly very important. They're sort of the code of life. And so biologists love to study genes. And if we're going to study them, we've got to call them something. We can't just say, that one. We have to be a little more specific so that we all know what we're talking about when we're writing papers. And fly people have gone sort of away from some scientific jargony stuff, and we tend to use sort of everyday words, but that's just what the genes are called. This one is technically longitudinal slacking, but people call her Lola, which I find really fun because that's my parents' dog's name. Right, so those are pretty fun names. If you're a scientist, maybe to you guys too. So today I'm going to tell you just about a couple of these genes and the story behind how they were discovered and named. So with that, I'll get started with the first gene I'm going to talk about, which is white, which is not a great, really fun name, but it is the first gene kind of ever. So this here is Thomas Hunt Morgan, who in 1910 was looking through all of his flies because he wanted to know how traits were inherited. At this point in time, we didn't know. We knew, obviously traits could be passed down from parent to child, but we didn't know how, and we didn't know what was doing that job. And so Thomas Hunt Morgan was on the hunt for a fly that looked different, right? He needed a trait he could study. And so he spent 2 years just looking at a bunch of flies under the microscope. This here is his little bottle of flies, still basically what they look like now. And after 2 perhaps boring years of work, he finds this fruit fly here, which has white eyes. Normally they have red eyes. And he calls this mutant fly white. And later, when he links this trait to a chromosome, we call that little chunk of the chromosome, right? That little chunk of DNA, that gene, we call it white. And so he sort of sets up this tradition of naming the gene after the mutant phenotype. Because this is basically, at this point, how we're discovering genes, is we're looking for mutants, we're looking for genes that are broken, we're figuring out where that is, and we're naming that little chunk white. So it's actually a little confusing because the protein encoded by the gene white is one that transports red pigment into the fly eye and makes it red. So it's a little backwards. So he did this in 1910. He trains a bunch of scientists, including one named Mildred Hodge Richards, who discovers this gene called eyeless. And you might imagine, based on the name, that the flies do not have eyes. Pretty obvious description of that phenotype. Right. So this is from a 1925 paper by Mildred and a collaborator named Esther Furrow. They're both at the University of Oklahoma, where, of course, Mildred does not have her own lab because of the patriarchy, but her husband does. It sucks. Anyway, she manages to do this beautiful work where Esther Furrow draws all these cross sections of the fly brain, and they sort of characterize what this mutant is doing, what the gene is doing, right? The loss of the gene. So this is the wild type, the normal fly. So you have the little brain here, and you've got all the columns of the eye. And it's a really nice picture. Now we would just take a picture of that, but someone sat down and drew that, and I think that's nice. And you can see in the eyeless mutants, that's figure 7, 8, and 9, you can see that they're lacking some of these columns of eyes, but they're also seeing some reduction in the size of the brain. And so this is sort of the first time they recognize not only does this gene eyeless have a role in eye development, it also seems to have a role in brain development, which is a little bit confusing because it is called eyeless. We know now that eyeless is a transcription factor, which is basically a gene that tells other genes to get made, right? So it tells it tells the cell, hey, you need to make these genes, you're gonna be an eye or you're gonna be a neuron. And I know it mostly, I use it in my work. We mark the expression pattern of eyeless in the nervous system with green fluorescent protein, and it labels this structure called the mushroom body, which is actually what I work on in my thesis work. So sort of a fun connection there. Right, so in the late 1980s, basically we're still finding genes by doing a mutant screen and then naming it after the mutant phenotype. So this is, the next couple are from Christine Uslan-Volhard and Eric Wieschaus, who are looking for genes involved in setting up the embryonic body plan, right? Because if you go from a single cell to a multi-cell, you need to make sure that's all patterned right so that you can be a real creature and not just an amorphous glob. So this here is the wild type, the normal embryo. You've got the mouth hooks up here, this is the tail, and they've got these nice little denticle bands that basically just have bristle. They have this hardened cuticle, right? So a lot has to go right for this to be a nice pattern. And so they name the gene sort of, it's a little bit more Rorschach test-like in that they look at the mutant embryo and they think, what does this remind me of? This first example, faint little ball, is maybe a bit literal. But that's why I chose it. I like it, right? There's a faint little ball of cuticle. You call it faint little ball. The next one is they have is Filzig. So Filzig means Felty in German. Christine Nielsen-Volhard, as you might imagine, is German. So I kind of like that she uses her native language to name this one. Basically, the bristles are a little shorter. You know, this gene is involved in signaling to tell those cells to bee bristles, and they do a bad job at it now that feels like it's broken. We also have ghost, which I'm a big fan of. Basically, it's just a completely undifferentiated mass of cuticle. All the cells are kind of the same. They don't know that they're in a segment, so it just kind of looks like nothing's there. Which is pretty fun. And then finally, this one, they kind of had to go more abstracted with mastermind. So this doesn't really look like anything. I would maybe call this, you know, scrunched up mass 3 or whatever. But this is actually the brain sort of poking out of the top, and it's sort of, I think, a reference to, you know, a cartoony mastermind with a big head, that movie Megamind. I don't know if that's relevant to anyone anymore. It was not that good of a movie. But they named it Mastermind, which is, you know, a little more creative than just faint little ball. Right, so far I've just told you about screens involving like the physical appearance of the fly or the embryo. But in the late 70s, early 80s, people were also beginning to be interested in learning mutants. So we wanted to find flies that were bad at learning so we could figure out what genes are important for learning, right? Like, what genes do neurons need to have so that the animal can learn? So the first one they find, they named dunce. So they do this fun little olfactory learning assay. Flies love to smell stuff. They do this little assay where they have all these flies in a start tube. They scooch this over to tube A, where they smell a smell, and then there's a little electric grid in there, and they get shocked. And flies, as you might expect, do not like to be shocked. So they do this a couple times, and you can see the normal flies, they measure how many flies stay in the start tube, and after you give them that smell and then shock them a couple times, more and more of them tend to stay in the start tube, although 40% still go into the shock tube, which is not great. Right, and then they do this test where they give them the tube, they smell the odor, but they're not shocked, but still most of them are staying away. Whereas the dunce mutants, once they're not getting shocked, they haven't learned anything about that odor, the shock odor, so they just go in there, smell around. They've not learned anything. And one thing I really appreciate about this paper is they actually give you some etymology about dunce and why they've chosen this. So I'm going to read it out. Dunce is a term of derision applied to followers of John Duns Scotus, theologian who died in 1308, regarded by 16th century humanists as an enemy of learning. And I mean, that's like a really great reason to name your gene dunce. Like it enhances the gene, right? Like without this gene, the flies can't learn. Therefore, the mutant is an enemy of learning. I love that they put that in there. Most people don't. They just name it something, no explanation. For example, William Quinn finds a whole bunch more of these learning mutants who also fail at this olfactory learning test. They smell a smell, they get shocked, they haven't learned anything. They still love the smell. So the first one he finds is turnip, and then he finds another one named rutabaga. I say he and the people in his lab. And then later there's cabbage. So I don't know if he was just really into vegetables or if this is sort of a reference to like a vegetative state, right? If this was applied to humans, it would really stink, right? Just because you can't learn well doesn't mean you're a vegetable. But because it's flies, you can kind of get away with it. So later on, we find some more learning mutants or a memory mutant, and we name it amnesiac, which doesn't really go with the whole garden theme, but is a really apt description of a fly who can learn but can't remember. But later in the 90s, one of William Quinn's students finds another mutant who has trouble remembering, and she names it radish to sort of fit in with the rest of them, which is, I think, a nice homage to sort of her training lab. Right, so with that, I've just told you about a couple of gene names. I hope you feel more positive towards fruit flies. What I'm going to leave you with is 3 general rules for naming a fruit fly gene, just in case that's, you find a weird one and you need to name it. So rule number 1 is it should be named after the mutant phenotype. That's the tradition. It's worked so far. You can go very literal and name it like eyeless, or you can sort of go more metaphorical and name it, mastermind. Rule number 2 is I think it's nice when you can sort of disambiguate it from the mutant phenotype, right? So like eyeless is hard because it also is in the nervous system where there aren't eyes. So it's nice to be able to sort of disambiguate it. So that's where I think like the vegetables really shine, right? Like radish is just a word. You can kind of separate it from the learning thing. And the third rule is I think it's nice when the name can sort of reflect the lab you came from or your nationality or something about you, right? So like, I like the sticking to the tradition of naming all your mutants after vegetables. That's kind of cute. Or Christine Nielsen-Volhard using her native language to name mutants. It's also kind of nice. So just in case any of you come across a weird fly, consider naming it something fun. Thanks.
Can you take questions? Emma will take questions if anybody has a question. And okay, I see a couple hands raised. Okay, well, then I won't say my question. I'll give you guys a turn.
Oh, right there. Right. So the question is, how do I think naming conventions apply to other scientific fields? Boy, that's a hard one. So obviously a lot of this doesn't work in a human context. So like we don't name human genes this. So eyeless is in fact in humans called PAX6, which is paired box 6, which is less exciting, but also is something that impacts human health. If you have a mutation in PAX6, you don't develop an iris and you have sight problems. So you don't necessarily want to tell a patient, hi, you have a mutation in eyeless. That kind of sucks. Naming genes is really hard because we don't know everything yet. So we can't come up with a name for everything. I know a lot of the early proteins involved in like DNA transcription and translation are just like elongation factor 1, elongation factor 2, elongation factor 3, and they're totally unrelated proteins. But we didn't know that at the time. We just knew, oh, we found a factor. And so it's a difficult task. And I think it's fun to give them these names that are more approachable and sort of reflect the scientists who made them. Yeah, in the back there. It is. These are flies that just live a little bit longer than the normal flies, under controlled lab conditions. Most flies I interact with don't make it that long because I use them for experiments. But under a controlled lab condition, they live a little bit longer. So they named it just I'm Not Dead Yet because they were waiting around for it to die. I think Out at First is a mutant in like embryonic hatching. So it's not a baseball reference, but yeah, here. I would love to know what you studied and if you found a gene. Oh boy. Okay, so the question was what I study and what I would find a gene if I named it. So I study how neurons in the fly brain come together and make partnerships, right? So like the way neurons connect is really important for the kinds of information they can hold and computations they can do. Boy, I think if I found a gene, I'd have to see what other related genes, if it's in a gene family, I'd have to name it something boring like DIP7. Right. But I think it'd be nice to start some sort of lab tradition of naming something, so if we found other stuff in the lab, we could sort of go off this tradition. Maybe flowers. We have time for one more question. Oh, back there in the dark.
Yeah, so with eyelids, what extent are the underdeveloped regions of the brain associated with vision?
Yeah, so the question was, with eyeless, what extent are the underdeveloped regions of the brain deal with vision? So the optic lobes carry a lot of visual information, so they're not getting a lot of input, so they're smaller. But the mushroom body that I showed in the middle, which is what I work on, is mostly olfactory learning and memory. And so that gets some visual input, but frankly, flies are mostly navigating the world through smell.
You'll take one more.
Yes.
I wanted to ask, the red eye versus the white eye, did that mean that it was just lack of pigment in the eye?
Yeah, so the question was, with the red eye versus the white eye, is it just a lack of pigment in the eye? Yeah, for the most part. So the gene white encodes for a transporter protein. So basically, when it's broken, the red pigment can't be transported into the eye, so they have white eyes. There's also a bunch of other eye color genes that are totally unrelated to that process that also impact eye color.
Thank you, Emma. All right. I think somebody should have named a fruit fly after one single fruit. Not a single fruit. Vegetables? They're not called vegetable flies. Fruit. But maybe that's why I'm not a scientist. That is probably the reason. All right. Thank you, Emma, so very much. That was delightful. And folks, it's break time. You have 10 minutes to go grab a drink, order more food. Top up. And Chelsea at the bar is dishing up delicious things. So please order something great and don't forget to tip. And we'll be back in 10. Thanks.
Talking away, I don't know what to say. I'll say it anyway. Today is another day to find you shying away. I'm coming for your love, okay? Take on me, take on me, take me on, take on me. I'll be gone. So needless to say, I'm upset, but I'll be stolen away. Slowly learning that life is okay. Say after me, it's for better to be safe than sorry. Take on me, take on me, take me on, take on me. I'll be gone. All the things that you say, yeah, is it delightful? Just to play my worries away. You're all the things I've got to remember. You're shying away. I'll be coming for you and you're like, take on me, take on me, take me, take on me. I'll be gone. Take on me. Take on me. I'll be gone. Take on me. Take on me. Take on me. How does it feel to treat me like you do? When you laid your hands on me and told me who you are. Thought I was mistaken. I thought I heard your words. Tell me how do I feel? Tell me now, how do I feel? Those who came before me lived through their vocations, from the past until completion. Will turn away no more. I still find it so hard to say what I need to say, but I'm quite sure that you'll tell me just how I should feel today. But if it wasn't for your misfortune, I'd be a heavenly person today. And I thought I was mistaken, and I thought I heard you speak. Tell me, how do I feel? Tell me now, how should I feel? Now I stand here waiting. Shout, shout, let it all out. These are the things I could do without. Come on, I'm talking to you. Come on.
And we're back. Folks, there are seats in the front if you'd like to come and aggressively watch Leanne give a talk. I see some of her friends are taking advantage of that. So if you want to be in the intimidation row, it's available. Filling up fast though. All right, so thanks everybody for hanging out and getting drinks. So Leanne Sue is going to tell us, she's going to be mad about this. She's going to tell us how she makes things go and then go faster. Is that accurate? Okay. So, but she can really do that because she is a PhD candidate in the Plasma Dynamics and Electric Propulsion Laboratory at the University of Michigan, where she makes things go. Please welcome Leanne Sue.
Thank you. Hello, everyone. So yes, I am Leanne. I'm a fifth-year PhD candidate, so really excited to do this because allegedly they're setting me free soon. So maybe won't be here next year. Maybe, hopefully, pray for me. So yes, today we will all be rocket scientists. I hope everyone here has seen this is Spinal Tap because we'll be turning it up to 11. If you haven't, thank you. So today we'll be talking about scaling Hall thrusters, which is what my research is about, to high current densities. So we will all become rocket scientists on this journey together. So space is the final frontier. This is the Starship Enterprise from Star Trek, if anyone doesn't recognize it. If you don't recognize it, go culture yourself. And we like space. And recently, there's been a big push about going to Mars specifically, which is in space. And to do so, we need spaceships like the Enterprise. And in fact, one of the things that NASA is really interested in is getting humans to Mars. And they kind of have, a game plan on how to do so. And what I'll be talking about today is this part of the spaceship. So the part that makes the spaceship go in space. And specifically, I'll be talking about the role that electric propulsion plays in getting humans to Mars. So what is electric propulsion and why is it good for Mars? So the challenge with conventional rockets, so like chemical rockets, which is what you think about when you see those big rockets not blow up, well, sometimes blow up, blast off into space, you see these big, plumes of fire and everything. Those are all chemical propulsion. And the issue with them is that they require a lot of propellant. And so when you need all that propellant to start off in space and then take us somewhere, it's really expensive to send all of it up because you need multiple launches with the abilities that we have now. And so more propellant means you need more launches to send everything that you need into space. Electric propulsion, however, instead of expelling a lot of propellant relatively slowly, electric propulsion takes a little bit of propellant and accelerates it so that it moves really quickly. And what this means is that electric propulsion, EP, is a lot more efficient than chemical. So we can use a lot less propellant, and a lot less propellant means a lot fewer launches. Fewer launches means we save a lot of money. So the takeaway here is basically EP takes more time, but a lot less propellant to get us to the same place, which is great. Ideally, obviously, we'd have something that is really light and really fast, but since we do not live in this beautiful fantasy world, we instead have to kind of make this trade-off between electric and chemical propulsion. So the two of the main things that we'll use to kind of characterize the performance of these thrusters is thrust, which is basically how much propulsive force we have. So how well you're able to kind of push yourself through space. And the other one is specific impulse, which is kind of a characterization of how quickly the propellant is moving out of the rocket. So it's the impulse provided per unit of propellant. And to kind of put us back on this little plot here, electric propulsion is low thrust, high specific impulse. Chemical is high thrust, low specific impulse. So how are we going to use EP to get us to Mars? And why haven't we kind of done it already? To reach Mars, we unfortunately need higher thrusts. So, you know, we kind of have to creep into the realm of chemical propulsion to get where we want to go. And the issue is that currently state-of-the-art Hall thrusters and electric propulsion in general is kind of capped at around 50 kilowatts, but we need to scale that up to a lot higher. And what we really need to do is increase our thrust density. So how much thrust we can produce with a given amount of surface area. So what should we use to get to these high powers? This has been spoiled. It's in the title, but bear with me because there's a lot of different types of electric propulsion. And over the past couple of decades, a lot of different types have been flown in space. And so this chart here kind of shows the different types of thrusters that have been flown. You can see early on there are a lot of resistojets, which is something a lot similar to, or more similar to traditional chemical propulsion. But in recent years, Hall thrusters have really kind of taken over the stage, and particularly with the launch of Starlink, which is SpaceX's internet provider. This scale you can see goes from 0 to 18. They launched like a couple thousand in 2020 and then a couple more thousand, I think last year, and then another few more. There's a lot of them out there now. So by and far, Hall thrusters have the most heritage in space. They're, you know, for better or for worse, SpaceX has made Hall thrusters the most common thruster in space. So what is a Hall thruster? This is a Hall thruster. So on the left here is a beautiful picture of a Hall thruster running. This is taken from my lab. We have a big vacuum chamber that we suck all the air out of to try to get it to feel more like space essentially, and then we run our thrusters inside. So this is a picture of one of them running. This is a horribly complicated diagram of what the guts look like, which I will be stepping through in a hopefully less painful fashion in the next couple of slides. So a Hall thruster. First of all, we have a voltage difference between our anode, which is positively charged, and our cathode, which is negatively charged. This voltage difference sets up an axial electric field that points from the anode, which is upstream, to the cathode, or in the direction of the cathode, which is downstream. Next, what we do is we have our cathode supply electrons, which are negatively charged particles. And then through our anode, we inject neutrals, so neutral, non-charged particles. When these neutrals bump into the electrons, if they have enough energy, they can ionize. And so the neutrals become ions, and then the ions are then suspect to these electric fields. And so our ions can be accelerated out of the thruster. And this is what we use to generate thrust. And the ions, because they're so much heavier than the electrons, when they get pushed out of the thruster, are able to push our spacecraft, albeit quite in little bits over a long period of time. However, because the ions are positive, the electrons are negative, our electrons are then accelerated towards the anode. But in an ideal world, we'd kind of have our electrons just stick around in the channel and provide fuel essentially for our neutrals to get turned into ions. So how do we keep the electrons there? What we do is we use magnets, which, as Jesse from Breaking Bad famously said, magnets, bitch, because honestly, I don't know how they work. I can tell you what they do, but I don't know how they work. So electromagnets set up a radial magnetic field within the thruster. And what that does is it interacts with our electric field, and then we get an azimuthal drift. And this is known as the Hall current. And basically, what this means is the combination of our electric and magnetic field is able to keep all the electrons trapped in the channel. And the reason that our ions are not trapped is because they're heavy, they're big boys. And so they're unmagnetized. Basically, they're not trapped by this same drift. And so they continue to be accelerated out, while our electrons in an ideal world are trapped in the channel forever, continuously providing ionization for the neutrals coming in. So that's how a Hall thruster works. There will not be a quiz later, don't worry. So the next thing we want to know is what happens to the performance if we try to increase the power? Like, why haven't we already done this and gotten to 200 or 300 or however many kilowatts and just, you know, send people to Mars already? And to understand that, we kind of have to talk about the performance of these thrusters with another metric known as efficiency. And specifically, we're talking about anode efficiency, which is basically kind of measuring the thruster on its lonesome without, why did it do that? Without thinking about the amount of flow going into the cathode or the power into the magnets. And this anode efficiency has multiple contributing factors essentially to it. The two I'll be talking about today are mass utilization and beam utilization, which are both scary words that aren't actually that bad, I promise. So beam utilization is kind of what we talked about earlier. It's how well those electrons are able to stay trapped in the channel. If you lose your electrons to the anode, then they're no longer available to keep ionizing your neutrals, so that's a loss. The other one is how much of your incoming neutrals you're able to actually convert into ions, and that's known as the mass utilization. And so what happens when we try to increase our current density, so increasing the flow into the thruster increases the current, increases the power. And so when we do this, when we look at the mass utilization, what happens is we have particles that are a lot closer together. And when they get closer together, they collide with each other more often. And so we get more neutrals getting ionized. And so our ionization efficiency goes up, which means our mass utilization efficiency also goes up. However, when we look at the beam utilization, which is much less agreeable, we see that we have this magnetic field, and ideally it keeps all of our electrons trapped. But if our neutral density is going up, then that means there are more collisions. And as there are more collisions, our electrons collide with particles that kind of yeet them back to the anode, and they're lost to us forever, and we can no longer use them, and it's very sad. So as we increase our current density, we kind of see this trade-off where our mass utilization improves, but our beam utilization gets worse. And so because of the worsening beam utilization, classically, these Hall thrusters, these devices, have been limited to thrust densities of around 10 newtons per square meter. However, this is classically, and as it turns out, the way that electrons behave in these devices is non-classical. This is a huge thing in my field. It is a ongoing area of study, but what we were interested in is, okay, if it's non-classical, what's the actual limit? And why is it 11? And so we used the H9, which is a Hall thruster that was developed between JPL, Jet Propulsion Lab, University of Michigan, and the Air Force Research Lab. And basically, they made multiple copies of this thruster that multiple institutions can use as a collaborative testbed. And our goal for this experiment was to increase the nominal current density that we operate at, so the kind of typical current density, by a factor of 10, and then see what the performance looks like. We used two different propellants, xenon, which performs better but is a lot more expensive, and krypton, which recently has kind of become the dominant one, again, thanks to Starlink. So this is what everyone came here for. This is a pretty sick video of our Hall thruster in the chamber. One of my lab mates mounted a camera in our vacuum chamber, which somehow did not immediately die. And this is as the probe arm is kind of swiveling around the Hall thruster, what it looks like. So we held our discharge voltage constant, and we went our discharge current. We swept from 15 amps, which is nominal, to 150, so a factor of 10. So check that one off. And one thing that my advisor always wants me to say is we were only limited by the power supply. If it went up to 175, we would have gone to 175. And hopefully we'll do that this summer with fun new power supplies. Here's more pretty pictures. This is what the highest operating power for each propellant looks like. Xenon didn't get to 150 amps because we melted some stuff and ran out of time. But krypton got there, so that's great. And so now let's take a look at the results. So first thing we'll talk about is, or the main thing we'll talk about again, is the anode efficiency. So basically, how well is this thruster able to perform? And what we notice is that the efficiency increases and then starts to decrease. This is for xenon. You can see it peaks at 50 amps. And for krypton, we have similar behavior where we see this increase before it kind of plateaus and starts to decrease. And the big takeaway here is that even though the efficiencies did decrease, they still stayed pretty high for this entire range above 50 amps. So all of these kind of atypically high current densities, the efficiency was still pretty good. So why does this happen? It seems like when we push past the traditional limit, there's not that bad of a drop in performance. So we were interested in looking at why that happens and also why we see this little peak. And to understand that, we can kind of revisit these two, beam and mass utilization. So again, how much of our electrons are getting lost to the anode and how much of our neutrals are able to be ionized. And what we saw is that the mass utilization, as we kind of expect, increases when we increase the current density, and then it stays at around 100%. And the reason for that is because, at lower current densities, we're not able to ionize all our neutrals. But once all the neutrals are ionized, there's not really much else to do. So now we just have a bunch of ions, which is great, but it can't continue to increase. With the beam utilization, we saw that it decreased throughout the entire range of currents as we kept increasing. And again, this was kind of expected. There's more particles in the thruster, so they get knocked around, the electrons get lost, and our beam utilization goes down. But we saw that this loss only ended up being about a 10% decrease, which is a little less drastic than a lot of kind of theoretical studies predicted. And the interaction kind of between these two efficiency modes is what leads to the behavior that we see, where we have an increase and then a decrease in the efficiency. So there's kind of these two competing effects that gives us an initial boost and then a decline kind of afterwards. So now we kind of understand why this is happening, and we have this really exciting result of the thruster being pretty good performance even at high currents. So how do we stack up to the competition? So first of all, let me introduce the competition. This is the Applied Field Magnetoplasma Dynamic Thruster. It has a really long name, which is one of the reasons I think it's less cool than a Hall thruster. It's an AFMPD, which is what I'm going to call it for the rest of this because that's too many syllables. And it's kind of the foremost contender for high power EP. The way it works means that it can get to a lot higher thrust densities, but one of the biggest drawbacks is there are not thousands of it in space. And it has other issues with its development, and it's much less mature technology. However, they're still kind of one of the biggest contenders when we think about high powers in electric propulsion. So here we compare it to what we were able to do with the H9 with our Hall thruster. So this is what our nominal current density is at our kind of baseline condition. And then all of these dots are different AFMPDs taken from a database at Princeton. And what we can do is we can plot what the thrust density of the H9 looks like on top of all of this. And what we see is that we're pretty much on that upper edge of when we compare to all these AFMPDs. So we're looking pretty good in terms of competitiveness with these devices, which up until now were considered one of maybe the only good contenders for high power electric propulsion. So when we operate at these high current densities, we can see that we maintain high performance, and we're pretty competitive with AFMPDs in this kind of up to 50 kilowatt range. And so kind of in conclusion, here's another fun video. This is the important part. So basically, our performance remained competitive with state of the art when we increased our current density by a factor of 10. Our efficiencies stayed pretty high. We saw what happened to the performance, and we were able to explain it. And then we saw that with an 8 times increase in thrust density, we're able to be competitive with AFMPDs. And this could have implications for Mars architecture. So how we design missions to Mars, especially with crewed missions. And again, we were limited by that power supply. There were plenty of other challenges. We melted some stuff, as I kind of alluded to. We'd like to be able to turn up the voltage as well as the current. And there's other challenges that kind of remain. But we're really excited about this. We think that the max thrust density is something we still haven't reached. And there's a lot more work to be done. But yeah, so that's continuing work in our lab. Maybe as we speak, I hope no one's there, but someone probably is. And I want to give a shout out to everyone who worked on this. This was a huge group project. I talk about it the most because I talk a lot, but everyone in my lab kind of worked on this. Won't name all the names, but they're all up here. And of course, our advisor and fearless leader, Professor Ben Jorns, led the way. The LSA machine shop at the University of Michigan was very nice when I sent them a piece of graphite at 5 p.m. and was like, please machine it by tomorrow. I need it. And then we had plenty of folks at JPL who were helping us and funding from Janus, which is a NASA institute. And with that, here is a sick logo that one of my lab mates, Tate, designed for the H9 muscle, because we essentially took a nice normal thruster and turned it into a janky muscle car. And yeah, I'll take any questions.
Any questions from lab mates?
Yes.
So when do you think we'll actually get to Mars?
That is a great question. The question was, when do I think we'll actually get to Mars? There's two wolves inside of me. One really wishes the best for humanity and thinks it's going to be soon, and the other thinks that progress takes a really long time. And I don't know, I guess, is the real question. This is only one of, I think, many challenges that we face when we're thinking about sending humans to Mars. So I think in an ideal world, a few decades would be my hopeful guess. Hopefully in my lifetime is more or less my answer. But yeah, soon-ish, question mark. Yeah.
So, assuming these advances are going to get locked in and be able to make, how big will the fuel tank need to be to get to Mars? Like, per kilogram or person, whatever unit.
Yeah, so the question was basically if these advances that we made are locked in and look good and permanent, how big will the fuel tanks be? Large is the short answer. And that's also kind of something we're looking at. Both xenon and krypton, the propellants we used here, are somewhat difficult to store in terms of, they need a lot of space. And so one of the things that a lot of research groups are looking at is alternate propellants, so things that can be stored more easily, like liquid metals, things like that. So yeah, definitely ongoing field of research. I can't give you an exact number, but big is the is the. Yeah, we like using ideal gases because it's easy to ionize them because of the way the electrons are. But yeah, there's definitely a lot of work being done on different types of propellants out there.
So if you're the captain of the USS Enterprise, what do you say when you put the whole thing on? Do you go like energize, engage, fly, ionize, magnetize, or my favorite in German is beschleunigen, change accelerate.
What would I say? Is your question? Okay, so the question is, what would I say if I was the captain of the USS Enterprise, which I wish, different lifetime maybe. What would I say when we start the spaceship? I punch it a lot. I think Sulu says that in the latest Star Trek, and then Spock tells him that the parking brake is on, and it's really embarrassing. When we turn, I think ionize would be pretty good. Ionize is a good one. A lot of times we kind of just, especially with new thrusters, if we don't know if they're going to work, we just sit there and then it turns on and we all go, ah, and then and then we hope it doesn't turn off, and sometimes it does. So yeah, ionize is a good one though. Yeah. Why did one melt is the question. Great question. As it turns out, when you dump a lot of power into something that is metal, sometimes things will melt. And so this is also something that's kind of an ongoing field of research. We're looking at how to keep them cooler, maybe with heat pipes or other things. There's also, if we don't kind of cover our the things that are electrically charged, if we don't cover them well enough, they'll arc to each other. That can also make things melt. So yeah, plenty of reasons that it starts to melt. There's actually, you can see in some of the pictures, things are glowing red hot. And that was one of the ways we could kind of tell when things are going well, because it's okay, it's bright red and nothing's broken yet. So this is good. Yeah, this is actually a big point of contention in my lab. Krypton is purple, and I will fight anyone who says otherwise. But yeah, so the different gases, when you ionize them, produce different colors just because of kind of the energy they let out. So yeah, the xenon is more of a kind of light blue, and then krypton is blue-purple. And as it turns out, all types of gases have all types of pretty colors.
One more, last one.
Yes. So the question is, how different would it be if Starlink was set up with chemical as opposed to electric thrusters? So first of all, it'd be a lot more expensive. And I think one of their priorities is keeping things cheap, which is also why they use krypton instead of xenon. So so one of the things with like those smaller satellites is the big purpose of the thrusters on them is to kind of just keep them in place, so they don't need a lot of thrust. They just need to be able to like kind of maintain their position. And so I think if they'd done that, everyone else in the aerospace world would maybe point at them and laugh and say, haha, you wasted all of your time and money on this stupid thing that is really expensive. Why did you do that? So I don't work there, so that's just my opinion on what would happen. But yeah, so I think it was a great idea that they used electric propulsion, especially because it's really good for my research.
All right. Very nice job. Thank you so much. And folks, we are again heading into, no, I went the wrong way. We are again heading, how am I doing this? We are again heading into break time. 10 minutes to refill, reload. So I send you all off.
I heat up, I can't cool down. Got me spinning round and round, round and round and round it goes. Where it stops, nobody knows. Every time you call my name, I heat up like a burning flame, full of desire. Kiss me, baby, let the fire get higher. I wanna reach out and grab you. Abracadabra. Abracadabra. You make me hot, you make me sigh. You make me laugh, you make me cry. Keep me burning for your love with the touch of a velvet glove. Abracadabra. I wanna reach out and grab you. Abracadabra. Abracadabra. I feel the magic in your caress. I feel magic when I touch your dress. Silk and satin, leather and lace. Black panties with an angel's face. I see magic in your eyes. I hear the magic in your sighs. Just when I think I'm gonna get away, I hear those words that you always say. Abracadabra. I wanna reach out and grab you. Abracadabra. Abracadabra. Every time you call my name, I heat up like a burning flame. Burning understand my intention. Weird science, elastic tubes and pots and pans, bits and pieces, and the magic from the handle make it weird science. Things I've never seen before. Behind open doors, talent and imagination. Weird science. Not what teachers said to do. Making dreams come true. Living tissue, warm flesh. Weird science.
Hi, everybody. We are back. We are back again. Thanks for hanging out, everybody. Okay, so please, we are going to welcome Kayla Zochowski. She is going to tell us about DNA, and she's a biologist, and her favorite Pokemon is Eevee. But that's not what this talk is about. It's about DNA. Please welcome Kayla Zochowski.
Well, you remember that. Hello, everyone. Thank you for coming. I personally am super excited to be back here at Live. So thank you, Live, for hosting. You're amazing. The last time I was here, which was many moons ago, I did present on my favorite scientist ever, Louis Pasteur. Was anyone here for that? Okay. All right. We have some people that have stuck it through. So yeah, LP is amazing. But today I won't be talking about LP. I'll be talking about DNA. So I'm a biologist, but I've dabbled in a lot of different ologies. I've done computational biology, virology, molecular biology, just say anology, and I probably haven't done it, but I've done a lot. So, but this is kind of my day-to-day. So these days I spend my entire day thinking about DNA, and specifically, what do we do with DNA and how do we sequence it and understand it? So we'll be talking about one tiny little aspect of DNA and how it's used, and that is in the context of ancestry. And I do have to thank Emma because she gave like a really nice intro. So we're all primed to be thinking about DNA and genes and all of those things. So it was a beautiful introduction. So DNA. DNA is our genetic code, and it's a double helix, and it really dictates everything about us. Emma said it earlier, you have DNA in every single one of your cells, and it helps us make who we are. We have DNA that says that we're human, but then we also have DNA that says that we're different humans. And this is what we care about today, is what is the DNA that kind of helps us understand potentially where we came from, what we're most like in terms of a geographical history. But let's just start with DNA, and then we'll dive into some of those details because it's important to understand this piece before we move on. So DNA, deoxyribonucleic acid, and Emma, again, stole some of my thunder. You a human. So if we kind of figure out where DNA is, okay, we're humans, hopefully. Then you've got, if we go one layer down, you've got tissues. So brain tissues, heart tissues, lung tissues, every tissue within your body is made up of cells. And all of those cells within each of these different cells have chromosomes. And then those chromosomes are essentially tightly wound DNA. So DNA is what we're interested in for right now. But you have all of these layers, all of these levels that go from DNA to all the pieces of us to being a human. So what is DNA and what is DNA sequencing? We also have these letters, and Emma showed this earlier. These are nucleotides. I'm not going to talk about them. What's important is that our DNA, all of the DNA in all of our cells, is made up of 4 letters. And it's the sequence of these letters that's important for establishing us as humans. Everything that I mentioned earlier, all of those tissues is made up of 4 letters and the sequence of those 4 letters in everything. Okay, so if we look at this double helix, we can say, all right, here are the letters that are making up this piece of DNA. And as Emma mentioned earlier, these can be genes, and the genes make up the genome, and you've got a lot of it. So DNA, chromosomes, here's, for example, a sequence of those 4 letters, just 4 letters over and over and over and over again. Okay, so DNA, this is just one tiny piece. We've got a lot of letters there. How many letters or nucleotides would you guess are in the human genome? Shout it out.
7 billion.
Okay. 12 million, 7 billion, 15 trillion. Okay. Normally people like really underball this. So this number is like, like this number is impressive, but the human genome is composed of 3.2 billion. So we had some over, some underestimates, but you know, you guys did pretty well. No Price is Right rules here. So 3.2 billion letters is what sequence of these letters makes us who we are. That's pretty impressive, but that's a lot of letters. And it makes sense because we are complex organisms. We've got a lot of stuff going on. We need a lot of letters to tell us how to be human. And then I felt like when Emma was talking earlier, I just wanted to do like a trivia question just to like see if people were paying attention. She did say that humans have 23 pairs of chromosomes, but how many do flies have? Fruit flies. All right. You guys are paying attention. Way to go. Gold stars for everyone. All right. So 3.2 billion. Now, if we're interested in actually characterizing this sequence, we want this sequence, and we'll talk about why. What do? Well, how do we get that? The first step is you need to get that DNA. So I did that whole progression of humans and tissues and cells and chromosomes. So you need to get that DNA out of those cells. All right. And this can happen in the lab. The next step is you manipulate the DNA. And you manipulate DNA because if you want to get the sequence, you can't just take a piece of DNA and read it like a book. There's a lot of technology that has to go into you being able to get that sequence out. This is a whole career. This is many companies just on this manipulation piece. And I'm just gonna kind of like wave it away and brush past it as if it doesn't matter. It does, but not today. And then next, after you manipulate the DNA, you're going to sequence the DNA or read. And again, we're just trying to figure out what this sequence of letters is, because that's going to matter for all of the things, all of the questions that we want to ask and answer. All right. So when we think about getting this sequence of DNA, reading a DNA sequence, there's a traditional method. And this method includes taking each of those letters, dyeing them a different color, giving them a fluorescent label, and then kind of building that sequence. And then when you reconstruct it, you can figure out what the letters are by the colors. Okay, pretty straightforward. Sounds pretty simple. That's great. But what you're getting out is one sequence from one piece of DNA. And how many letters do we got? 3.2 billion. So this is going to be a really long process if you're trying to do 3.2 billion. So what the field of sequencing and kind of biology, molecular biology in general did was come up with next generation sequencing. It sounds very fancy. But what next generation sequencing means, or NGS, is just that instead of looking at one sequence, you're going to be looking at millions of sequences at one time. So it's kind of just making everything high throughput. That's all next generation sequencing is. That's it. Here are some instruments that do this next generation sequencing. And I kind of have a variety here. There's lots of different instruments that can do this. But what I want to highlight is that you have some instruments that can fit in the palm of your hand. This can read a sequence. This can read a DNA sequence. You have some that are kind of like a little benchtop instrument, and then you have some that are as tall as a human. So there are lots of different ways and lots of different scales at which you can sequence DNA. Does anyone remember the Human Genome Project? All right, we got some hands up. Yeah, big deal, right? So this is back in 2003. They were trying to sequence the human genome. They wanted to know what the human genome is. 2003 was several years of work, well, probably more than that. But we've got, they published the human genome. They're like, all right, we've got the human genome. Who wants to guess how much money it took them to get the human genome in 2003, 2004? Okay, wow. Why is everyone doing trillions today? There's so many trillions being thrown out. All right, well, it was a lot of money, but it was 3 billion. So we're still in the billions, folks. We're in the billions. But if we think about that, that's almost like $1 per letter, which is interesting. But so things used to be really, really expensive. But with the advent of next generation sequencing, we now have a huge drop in the price of sequencing. So it used to be, to get your human genome sequence, the Human Genome Project was $3 billion. But even after that, it would cost you $100 million to get your genome sequenced, which is a lot of money. If they had just waited until next generation sequencing came along, well, now we're looking at less than $1,000, which is, a little bit more palatable. All right, so this is important because what this means is that having this decrease in price means that we can do things like what I will be talking about today, which is spitting in a tube, paying a couple $100 and getting some information back about your DNA. And then just because it was May the 4th recently, I saw this from the National Human Genome Research, and it's essentially, even though they're saying the human genome was published, in 2004, that actually wasn't even all of the genome. It was about 92% of the genome. So I love this because it's like the human genome sequence is finished, and it's like all 3 billion bases, right? All 3 billion letters, all 3 billion letters, right? So it's actually not until very recently that the entire human genome was actually sequenced. And there's lots of reasons for that, and I would be happy to talk about it later. All right, so why would we want to sequence DNA? The talk today was kind of advertised as ancestry. We're going to be focusing on this. And then depending on how long I talk, if I have just like 2 minutes, I'm going to touch on some other topics such as oncology, medications, and infectious disease. And this is just a subsample of the reasons why we would want to sequence DNA. But let's focus on ancestry for tonight. Who has gotten an ancestry test? Okay, I see about maybe a dozen different hands up. Who did Ancestry? Ancestry.com. Okay, some hands. And who did 23andMe? Okay, and then who did something else? Okay. All right, so we've got kind of a split between 23andMe and Ancestry.com, but there are a bunch of other options out there. All right, so if you've done one, what you know is that you spit in a tube, you mail that tube, and then you get data back. But what we care about is what is that data? What data are you getting back? You sent your spit off, what do you get back for that? And your couple $100. Don't forget about that piece. So here's you spitting in a tube. And here is the information that you would get back. So what you want, what you're interested in when you spit in the tube and mail it off. And I don't know why I'm like so fixated on the spit in a tube thing, but I just think it's funny. So like you spit in a tube, you send it off. And so what you're interested in is information about the probability that your ancestors came from a specific geographic region. So you've got some probability or some percentage, your distribution. What is your genetic makeup? And this is the information that's pretty cool to see, right? So we're going to talk about how you go from this, spit in a tube, to this information. That's cool for you. Okay, let's do it. So we talked about earlier the process for getting a sequence. We started with extracting the DNA. So why does it work for you to spit in a tube and mail it off? Science. Yes, science. Like we talked a lot. Yeah, go ahead, just shout it out. Exactly. Yeah, I heard it from two different people. Skin cells. So there's skin cells inside your mouth, kind of in your saliva. And we talked about how your DNA is in all of your cells. So when you spit into a tube, there's skin cells in there. And when you ship it off to a company, they can extract the DNA from those skin cells that were in your saliva. So they get your DNA. Okay, cool, great. Then they do all of the sciencey things. They manipulate it and they sequence it. Now, how many letters are there in the human genome? 3.2 billion. Now, when you send off your DNA, they're not going to actually sequence all 3.2 billion. If you remember that diagram, like it used to cost $100 million, and even now it's like $1,000 and you didn't pay $1,000 when you sent off your spit. So what the companies do is they actually focus in on certain genes and certain sites within those genes. So you've got some chunks of DNA here. They're only interested in this site and this site and this letter and this letter and that letter. That's all they care about. And the reason is because we actually, let's see what I've got next. Oh, yeah, I've got this number. We know this number now. The next question is, how many of these sites do you think that they're interested in? And then I'll do my punchline. 20. All right, we got 20. 100,000. Okay. We're getting close. Okay, it's not in the trillions. So the answer is 300,000. So this is the number of letters that they're going to be looking at. And the reason is because we're all human, again, hopefully, and we share about 99.8% of DNA with each other, because all of that DNA goes to telling us that we're human, that this is a heart, this is a brain, this is muscle tissue. All of that is saying that we're human, and it's really only a tiny percent that makes us different from each other. And that's the piece that's going to be important when you're trying to understand your geography and what your ancestors might, where they might have been located. So 300,000 different sites, less than 0.1%, and this is going to be able to help us differentiate geography. And I kind of made that little like snide comment earlier about like, oh, it's $1 per letter. Luckily, NGS is here, or else this would be a $300,000 test. So just thinking about money here, and how impactful NGS can be in terms of making this accessible to all of us. All right, so let's think about how this works. We talked about 300,000 different letters are going to be getting sequenced, but okay, great. What do you do with that? If we kind of just do an example, we've got 5 different genes here, and let's say these are the letters that are going to be sequenced that are important to have that information for. Okay, perfect. These letters, we talked about how they were nucleotides earlier, the A, T, C, and G. These are called single nucleotide polymorphisms. And if we kind of break that down, poly means many and morph means form. So all this is a SNP. A SNP is just essentially a site where you can have many forms of a nucleotide. But these SNPs are, I don't know why I'm using air quotes, they're called SNPs. These SNPs are what is being sequenced. These are the letters that are important because they are the letters that are different between all of us. And I'll be using SNPs throughout the rest of the talk, but we're really just talking about sites where nucleotides might be different, letters might be different. Okay, so how do we get from this to information? Well, we have a database. So here I've got a table. I've got these 5 genes depicted, and we've been this is an example, I've been by continent, but we've been by continent what we expect to see, which letter we expect to see for each of these regions. Okay, great, we've got our table. So now what we want to do is figure out this person, which letters match, what the composition would be. So if we just highlight what letters they have here, where it falls within the table for each of these genes. Okay, perfect. If this was as simple as this person would be 40% American, 40% Asian, 20% European. All right, great. You've got your ancestry test. Woo! But of course, this is a simple example and it's not this simple. How many sites did I say that we're going to be sequencing for this? 300,000. So we don't have 5 rows in our table. We have 300,000 rows in our table. And like, if you want to try and open that Excel document on your computer, I mean, good luck. But in addition to that, we don't have 4 columns. We have hundreds of columns, because what we're really trying to do, well, we as in what science is really trying to do is pinpoint a geographic location. So all of these kind of color blobs are representing a different population that you could be matched to. And so you now have hundreds of columns and 300,000 rows, and that's a big table. Now, the next question would be, okay, I gave an example table, but like, how do you get that table? Where does that table come from? And the answer is The ancestry companies. So they make that table, they make that database. That is part of their business and part of what is important for you to send your spit to them as opposed to, some other random lab. So they create the databases, and these databases are based off of self-reporting, family trees, historical records, and then math. And so, they make this database by trying to really understand a family that might say, okay, I know that my family's been in Italy for 8 generations. And then the ancestry.com is like, okay, cool, let's check this out. And they go and they look at historical records and they say, you know what? Your family has lived in this tiny region of Italy for 8 generations. Then they take your snips and they're like, all right, adding it to the database. So everything you get, all of the information that you're getting when you get data and when you get these probabilities is because there's some evidence that would link those particular SNPs, those 300,000, to a geographical region. Now, I'm sure the skeptics in the audience right now are already like, well, can you really trust the self-reporting and the family tree and the historical record research? And the question is a good one. I mean, we don't know what reality is. let's not get too crazy, meta here, but we don't know what reality is. All we know is what we can piece together based on data, based on science, based on stories, based on records. And this is what's going to create that database that you eventually compare to. Okay, so you got your database, and then the next thing is how is your DNA matched to that database? Okay, I've got 3 things. First is Sensei Dave. So Sensei Dave used to be my mentor for competitive Scrabble. He's great. He's phenomenal at it. And I was just telling Sarah, I was like, if I get invited back, maybe I'll talk about competitive Scrabble. Who would be interested in that? Oh my gosh, you guys are amazing. All right, everyone yell bingo. All right, and if you don't know why we just yelled bingo, you should come to the competitive Scrabble talk. All right, so Sensei Dave, great at competitive Scrabble, but he also was a programmer at Ancestry.com for almost a decade. And so he does a lot of the programming to help make the math to match your data to a database. We've also got inheritance patterns. So the DNA that we have, 50% of it came from our mom and 50% came from our dad. When you look at this, kind of when we're thinking about, let's say this is you, and sorry, the colors aren't super visible, but you're essentially a mosaic of the colors or the genes or the DNA of your parents, of your grandparents, et cetera. So when they're trying to understand what you might be, they're kind of taking little blocks of each of these pieces of DNA and comparing it to the database. And they take the next block and compare it to the database. And they do that for the whole genome. And that's because, as you can see, different parts of your genome have different origins. And then there's this thing called hidden Markov models, where they're modeling this over and over and over and over and over. And I think this says 1,000, but they might do it more than that. I'm not going to quiz you on that number. It's one of the numbers we won't talk about. And then at the end, they get a probability that this is the most probable composition of your SNPs compared to a database of SNPs. All right, so I've got a few questions for you. Can your results change? Yes. All right, I'm hearing a lot of yeses. Certainly they can. The reasons are going to be, well, you have more data in the database. There's more research that's been done, or there's different curation that applies. Got a time check. How much time do I have? Like 2 more minutes? Yeah. 2 more minutes. All right, more data for the database, and then you can have more math by Sensei Dave to kind of really get those algorithms optimized for doing that probability. Can your DNA change? All right. Yeah, this is kind of a trick question. Yeah. The answer is yes, but not really in a way that would ever impact your results. This is because you can have mutations that occur, but these mutations would occur in localized things. And again, we're spitting into the tube, so we're kind of getting cells that are represented in all of our bodies, and a mutation that occurred later on wouldn't be a part of that. So trick question, sorry about that. Then final question, can my ancestry results be different from my siblings? Yes. All right. You guys are amazing. This is because we're going back to that 50% mom and dad. So if we have grandparents, they get jumbled up to make parents. And then you can see that all of these siblings have a different composition of their parents. So they're getting 50-50, but it's not the same 50-50. So if these three individuals took an ancestry test, poor Gerald wouldn't have whatever green is. It'd be 0%, but everyone else would have that. And he'd be like, was I adopted? And then they'd be like, no, you weren't. All right, so this is really the end, the conclusion. So hopefully you have a little bit better understanding of how when you spit in a tube and you send it off, you can get information. I'm not going to comment on, kind of which database is best. I mean, each of these companies have a lot of documentation, so feel free to kind of sift through. This is like a 23andMe report. This is like an ancestry report. They're all going to have kind of a little bit of different nuances between them. And yeah, I didn't get to, I talked too much, so I didn't get to chat about these other topics, but I'd be more than happy to talk about it after the fact, because there's really so many ways that sequencing DNA can be impactful for understanding ancestry and for other things like oncology, infectious disease, tracking pathogens. I didn't get to tell my favorite watermelon PSA, but if you're interested, come talk to me about watermelons. Washermelons. All right, thank you. Oh, let me go to my thank you slide. There we go. 2 questions. We got one over there. Oh my gosh, thank you for asking. Watermelon. It's an infectious disease thing. So who here likes watermelon? Yeah. Who washes your watermelon? Not as many hands, So what happens is I visit a lot of public health labs that do sequencing. And so what they told me, which is my favorite PSA, well, favorite and least favorite, is that you pick up your watermelon from the store, and there's like all this like lovely bacteria on the outside. And you're like, I don't care. I'm not going to eat the outside. But then what do? You take a knife, you cut into it, and now you've moved all of the outside bacteria into the inside, which is a nice sugar-rich environment that the bacteria will love. So they say that the foodborne outbreaks in the summer are the most from melons. So wash your melons. Wash your melons. Thank you for asking. Yeah. Yes. Yeah, great question. The question is about when we talk about these databases, how is the reliability for people of color versus Europeans? Because there's going to be a difference in how reporting happens, what historical records are available. And that is such a good point that the scientific community is trying to work on. There's a lot of work on adding sequences, on adding to databases to represent minorities, because even in a lot of health contexts, the healthcare that we do in terms of sequencing is all based on a European reference set. So something that I don't know about the reliability, but but it is something that the scientific community is very aware of and trying to fix. Okay. All right, there. Yeah, I figured I was going to get this question. The question is about, do these companies share your genetic data? The answer is that there's lots of paperwork that you probably sign when you send off your spit. Maybe read that paperwork. There's different settings for each company. There's different rules about what they do. Sometimes they will anonymize your data and share it with use it in their database or share it for research. Sometimes they won't. It's very up in the air. And this is such an important question in terms of how we think about data and our biological data. What they do with the rest of your spit when you send it off. Some of the companies will destroy it, some won't. So yeah, read the fine print for this for sure. Okay, one last question back there in the booth. Oh, what do I do? I am a sequencing specialist. No, I work for a sequencing company, and I essentially am a bit of a consultant. So if anybody is interested in sequencing, I'm the person you call and I say, okay, here's how you extract your DNA. Here's what you do to manipulate the DNA. Here's how you sequence it. Here's how you analyze the data. And I know a lot about sequencing. So that's what I do. Yeah. Thanks for asking. Dang. All right. Thank you all.
Thank you so much, Kayla. Amazing. Thank you. Thanks for asking who she is. A proper host would have done a proper bio, but unfortunately you have me. So, but if you think that you would like to do Nerd Nite, if you think you've got a story in you, if you have a favorite Pokemon and you want to tell me about it, then you should check out the Nerd Nite website, which I did not add to this slide. But if you Google Nerd Nite Ann Arbor, you'll find it, no problem. And you can shoot a proposal to us and like tell us what you want to talk about and why you are a good person to talk about it. And we'll talk and get you on the schedule. It's great. We're always looking for speakers, and there's people in this very room, John, who have done the very same thing, have emailed me and then gotten on the schedule. So it can happen for you too. Thank you so much to our amazing speakers, to Kayla and to Leanne and to Emma for coming out and for their expertise. One more round of applause for them, please. Thank you, guys. And then I must direct your attention to June 21st at Summerfest will be our next Nerd Nite. It's going to be great. We have a bang up slate of presenters. One will be a one-man band with a ludicrous, just invented instrument. And I cannot explain to you what it is or how it makes the sounds it does, but it makes so many sounds. You got to come. You have to come. It is indescribable. Okay, so it's going to be great. We will see you soon. I hope June 21st. In the meantime, thank you live. And everybody, thanks for coming out.